Hi, this is Randy Carden from Iowa City. Thanks for presenting this. My question is that I know in recent years the macula has been shown to be much more involved than people realize in glaucoma, especially when you do 10-2 testing and ganglion cell OCT testing. But optic nerve drusen most of the time spares the central 10 degrees. It'd be unusual for it to affect the central 10 degrees. So even with a multifocal VP, if the damage is occurring outside of that area, do you think there's still enough signal to noise to pick it up with evoked potentials?
Yeah, evoked potentials are heavily biased towards the central visual field. And so that's a challenge. So we work very hard on techniques that improve the signal to noise ratio of these sorts of measurements. And the hope is that despite this, that there may still be enough signal to noise ratio to be able to work on the periphery. And we're cautiously optimistic, but it's a big challenge. And it's one of the downsides of the evoked potential in some senses, that it's so foveally dominated. And it's also harder to record responses from the upper visual field just because of the geometry of cortex. Right. So that's another weak spot in the technique.
Thank you.
Randy, I just add one comment to that, which is that among the glaucoma patients that we've been studying so far, there are quite a number of patients who have mild field defects that are much more the peripheral arcuates that would be found on a 24-2, but completely missed on a 10-2. In fact, I'd say there's probably a plurality of them. So the aggregate data do still suggest that we're detecting changes or differences on these current full field or hemifield approaches so far. So we haven't specifically studied the question that you're asking, but if I had to guess based on the patient population that we've been studying so far, I think we do have a good shot at being able to detect those more sort of 24-2 zone defects.
Joyce, have you or anybody been doing 10-2 fields in drusen patients just to see if there is some effect that we're not picking up because the spacing of a 24-2 just doesn't sample it very well?
We haven't consistently been doing the 10-2s, but the idea of the Optic Disc Drusen Center is that we'll take willing participants, and measure everything. So hopefully we'll be able to answer that question.
Have you seen much ganglion cell loss within the central 10?
You definitely see thinning of the macular ganglion cell complex. But in terms of correlation to function, for within the central 10, I think we haven't really done that structure function comparison to be able to answer.
But you are seeing thinning.
Oh, absolutely. And it's a pattern that's very typical for optic nerve head diseases like glaucoma or papilledema. You get these temporal thinning first. And it's very nicely corresponding to the pattern in terms of a 24-2 visual field test result.
Hi, this is Jeff Odell from New York. I have a couple of questions. How does cortical folding and the amount of the visual cortex that is on the surface affect your recordings? How long does it take to do this? And are you using a best channel kind of approach?
Yep, all good questions. So the first cut on that is the polarity of the response is for the early part of the response is the opposite sign in the upper and lower visual fields. So if you present, like I showed you, data from both of them simultaneously, you actually miss one of the early peaks that you can see if you do hemifields. So it's another reason to go to hemifield stimulation, is that you can see this very early visual cortex peak more clearly. The other issue is as you go eccentric, the visual field map goes deeper. So that's one of the reasons we have more trouble recording from the periphery along with magnification, is the tissue is actually further from the electrodes. And that's especially true for the upper visual field. And I think you had another question.
How much time are you spending doing this?
So typical measurement per eye, we would do 8 to 15 10-second runs per eye.
So
to
that's
make
much faster than multifocal. And are you using a best channel approach?
Yes. So we do a component decomposition technique. So it's effectively finding in a data-driven fashion what the optimal weighting of the channels are, so you don't have to pick a best channel. And our prior work on this has shown that this spatial filtering approach, which uses multiple electrodes, actually has better SNR than the best channel. So it's another thing in the background in the methods development that I didn't have time to get into.
Thank you.
We have another question from Mr. Eric Geyer. Are there any changes in VEP that occur as a result of repeat testing, even in normal individuals?
There have been studies of perceptual learning using evoked potentials. So this would be practice effects. So they tend to be small and kind of hard to measure. So in general, what people have found with repeated testing is that test-retest reliability is quite good, but between subject differences are quite large. So that's one of the issues with any of these electrophysiology techniques, is the between subject differences are quite large, but the within subject test-retest repeatability is much more stable. So that suggests that maybe one of the best applications for these techniques would be looking at progression because of the relative stability on repeated testing.
Thank you.
Alf, this is Shannon. I see AI is such an exciting field moving forward. Can you let me know the feasibility of doing this? How does the protocol work? How long does it take to image one eye, both eyes?
Yes, the optic disc, because it covers a small area of the retina, then it's a lot more feasible than, say, image a maculopathy. So we could image each eye in, say, maybe 10 minutes.
And how still do they have to sit for it?
Traditionally, we ask people to use a bibar, but we've done that with a chin rest if the patient or the study participant is engaged enough. But it's, the bibar is not a deal breaker. It's desirable, but it's not a deal breaker. We've imaged children as young as 8.
Is it similar to the OCT where if you move, you then pick up from where it last was imaging, or does it start over?
Yeah, we capture, yes, you can start capturing, and then you don't have to be still for 10 minutes. You have to be still only for about 10-15 seconds at a time. And if each 10 or 15 second scan is affected, say, by motion, then you can always take it again.
There's a question from Susan Altman that says, do you find inattention to be a factor in your results?
To find, yes, we have people that feel comfortable in the room that is warm and dark, and sometimes they fall asleep. So we tend to engage with the study participants and talk to them and take frequent breaks and talk about non-disease related topics to keep them engaged. But it's not a big factor.
Dr. Norsha, there's a question from Jessie Gale that says, are the VEP abnormalities purely amplitude, atrophy, and cell number, or are there other signal changes? In either glaucoma or optic dysdrusen, is there suggestion of cellular stress or axoplasmic impairment or reversibility in VEP?
There's a pretty strong latency we effect. That is, the response gets smaller and slower with glaucoma severity. We don't have enough data yet in optic dysdrusen patients to know whether that's true or not. But that's a very, very clear.
Actually, related to that, I was wondering if Jeff Odell or people in the audience, has anyone looked at VEP studies in optic dysdrusen?
On rare occasion, we have, they kind of look like glaucoma and ischemic optic neuropathy, as distinct from MS. We didn't find a very robust change in timing. We found more a change in amplitude on multifocal VEP.
Yeah, I think that's going to be one of the questions of whether these, by playing around with the stimulation parameters like the increment decrement in particular, whether that's differential. And it could cut either way. It could be that we find that off ganglion cells are always hit in an optic nerve disease. And then that pushes you in the direction of saying, what's funny or different about the off pathway than the on pathway that makes it more vulnerable? So like, it could go that way. Or it could be, oh, this only happens in 2 of the 3 major optic neuropathies. So at least you've got some, a bit more, you have another parameter in which you could differentiate different optic nerve diseases. And so either way it goes, I think it's going to be interesting.
Could you envision using a combination of multifocal VEP with the on and off pathway difference?
Sure. Sure. It's easier for me to think about designing the selective stimulation not in the binary M sequence approach. It's a little harder to make that asymmetric. So we're using sawtooth stimulation in the steady state VEP. So there, the multifocality would come about from using multiple different temporal frequency tags. And other groups have pushed this up to 8. So I think that's a feasible 8-sector probe. Multifocal at the level of 8 sectors is probably within reach.
So Tony, with you and Jeff's study on glaucoma, I was curious if you are able to measure changes cortically in areas where the visual field defect may not be present. On the standard visual field testing.
Yeah, I'm not sure how one would do that. I mean, I think it's really interesting to think about cortical adaptations. So you could say, oh, I know there's the visual field losses here. Has there been compensation peripheral to that? I think that's your question. It could be, is the loss in V1 somehow made up for in higher level cortex? So like if you looked at early visual cortex responses and higher level cortex responses, do you see the same level of deficit as if it's just being passed on from the retina? Or potentially if cortex is really good, it could say, oh, I've got a deafferentation and I'm going to kind of do my best to kind of make up for it, cover it over a bit. And that, I don't know if that happens. I don't know any examples of it having happened, but we're, I think, in a position to look for that sort of thing.
It's a super interesting question because, you know, we know throughout the nervous system that you can lose you know, in different diseases, anywhere from half to 80 or 90% of your neurons without detecting really significant deficits in that neuron's responsibility, let's just say, in the phenotypes. And so, you know, is that because of redundancy at that local level, or is it because of ability to fill in later and make better do with whatever information is coming through? It's a really cool question.
There's limited study on other optic neuropathies like neuromyelitis optica and optic neuritis using MRI. And it seems that this cortical adaptation at a higher level could occur relatively quickly, meaning it could be potentially measured within a short time. And so it seems that the cortex is quite dynamic in terms of the ability to adapt and compensate. And the question is whether we could actually measure that in a way that would be of value to visual experience.
Well, yeah, I think one of the things we didn't talk about is the potential for using functional MRI, which is also on the table with Justin Gardner's group.
Mm-hmm.
And so one thing you could do perimetry with functional MRI, and you can do it in multiple visual areas at the same time and ask whether the field maps are equally degraded or whether there's relative compensation in a parametric fashion.
Mm-hmm.
Great. Well, thank you, everybody. This has been a nice session on human imaging and biomarkers in optic dystrophin. We're going to take a 20-minute break. We'll be back at 3:15, and we will then start our next session on treatments in optic dystrophin and other optic neuropathies. So thank you, everybody, for joining us for this last hour and a half.